Hey everybody, let's talk about CRISPR technology. Now in the previous video I described how the type 2 CRISPR system was the most commonly used in biotechnology and research. And one of the most common things to do with the type 2 CRISPR system is to mutate genes. And at the end of this video we will see another use for this type of technology, not just mutating genes, but also, changing the sequence of a gene in a specifically desired way. So how would we use CRISPR technology to mutate a gene? Basically, and we will talk about specific ways in lecture 24. But basically what we need to do is get inside a cell. So let's say we want to mutate a gene in a eukaryotic organism. Well, we need to get Cas9 inside a cell and a guide RNA inside a cell. So in eukaryotes, we need to get these in the nucleus too, right? So bacteria don't need to worry about that. The viral DNA that the natural Cas9 CRISPR system is targeting, the virus is just in the bacterial cell, which doesn't have a nucleus. But if we want to engineer a eukaryotic genome, we need to get these inside, not only inside the cell, but inside the nucleus. So assuming we can get those in, we just need to get those two in, and then we can mutate a specific gene. So get these into cell/nucleus. Okay, so assume we can do that. We'll talk more about that in lecture 24. In nature, the guide RNA is made up of two RNA molecules, right? So in nature, we need a tracer, or the bacterial cell needs a tracer RNA and a CRISPR RNA. So in research or biotechnology, to engineer genomes, scientists have figured out we can use one RNA molecule. So a single guide RNA molecule. Sometimes this is abbreviated as gRNA. Okay, so how does that work? Well, let's just take a quick look of, comparing the two systems, the natural system and the, let's say, biotechnology system. So let's say this is Cas9 right here, and this is the natural system over here, and this is the biotech system over here. Now I'll diagram this, I think, a little differently than I had in the previous videos, because we should pay attention now to the 5' and 3' ends of these molecules. So let's say this is the CRISPR RNA of the natural system with the spacer sequence that matches the viral DNA in the repeat sequence that's repeated in the CRISPR array. That's the 5' end, this is the 3' end. And then this is the part of the tracer RNA, the 5' end over here, that matches the repeat region of the CRISPR RNA. It's complementary. Then here's the part that doesn't match, but forms into some sort of hairpin secondary structure that is recognized by Cas9. And this is the tracer RNA. So Cas9 binds the tracer RNA binds the CRISPR RNA, and this part of the CRISPR RNA is used as a guide to find viral DNA sequences. In biotechnology, scientists have figured out that, we can use a single RNA molecule that looks just like this tracer RNA, CRISPR RNA sequence. So we can synthesize a guide RNA molecule of about 80 nucleotides. And where do we get that 80 nucleotides? So let's assume all of these are about 20 nucleotides long. 20, 40, 60, 80. Okay, so single guide RNA of about 80 nucleotides long, and it looks something like this. Okay, so this would be the sequence that would match whatever gene we want to mutate, about 20 nucleotides long. And then that would go, this is an RNA molecule, it's a single-stranded RNA molecule, just like these, single-stranded. So instead of ending right here, what it's going to do is going to curl around, and we're going to have another sequence right here that's complementary to this right here. So we got this hairpin now. So in the natural system over here, there's two strands that are interacting by hydrogen bonding. So here, this is one RNA strand, one single-stranded RNA that's interacting by intramolecular hydrogen bonding. So this would be intermolecular, two molecules, hydrogen bonds forming between two different molecules. This is intramolecular hydrogen bonding. The hydrogen bonds are forming between nucleotides of the same RNA molecule. Okay, we can just continue that up here. And essentially it looks just like, same sequences that are found in the tracer RNA and the CRISPR RNA, with some extra sequences right here to link these together. Okay, so that's the guide RNA. And it's much easier than, or it leads to increased efficiency of Cas9 mediated cleavage of the target gene when you're using a single guide, because you don't have to worry about, is Cas9 going to find the tracer RNA, and is the tracer RNA going to find the CRISPR RNA? Cas9 just has to find that guide RNA. Then it can function, as long as it's in the nucleus, right? So let's say we wanted to mutate a gene. What would we do? Well, we would need to design the guide RNA, and we could order the guide RNA from a biotechnology company. Let's say we wanted to inject Cas9. We could order Cas9 protein from a biotechnology company and order a guide RNA from a biotechnology company that would synthesize it for us. And so let's say this is our guide RNA of about 80 nucleotides in length. So these 60 are going to be invariant. They're always going to be the same for every guide RNA molecule we want to order. For every gene we want to mutate, these 60 nucleotides over here of the single-stranded RNA are going to be identical between all the different guide RNAs. This is going to form that hairpin structure that, Cas9 recognizes. And this right here, well, this is going to be complementary to the gene, complementary to target gene, the gene we want to mutate. Okay, so how would we pick a sequence? So let's go over that, because it will help you review some concepts we covered in the previous video. And if you were confused, it might help you be less confused if we actually pick a sequence for targeting by Cas9 mediated mutagenesis. So I've put some sequences in here. So we have let's say this is a sequence of a gene. Let's say it's an exon coding sequence inside a gene, and we want to mutate it. If we mutate it, maybe it will create a null allele, and so we can figure out what that gene does. So now when we're looking for the bases to target, well, remember what we need to do is we need to look for a PAM site, right? Because Cas9 will only cut the target gene if the guide portion, or the complementary part of the CRISPR RNA is identical to the target gene, but it's also next to a PAM site. So what is the PAM consensus sequence for Cas9? And I should say this is a Cas9 from a specific species of bacteria. And that Cas9 prefers 5' NGG to be the PAM sequence. Other Cas9s from other bacteria may use a slightly different sequence, but NGG is the one we're interested in. It's the one that is the it goes with the Cas9 that is most commonly used in labs around the world. So what we need to do is look for a 5' NGG, and we can look up here. Do we see 2 GGs anywhere? Oh, I see some right here. So this thing right here, this is a suitable PAM sequence. And so what we would need to do is just count 20 nucleotides this way. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay, and then this thing right here, so this sequence, this is what we would, tell the biotechnology company to use when it's synthesizing the guide RNA. And we'll call this is called the guide portion of the guide RNA. And that makes sense, right? Because it's guiding Cas9 to the gene. So the guide portion right here, it stops immediately before the PAM motif, the PAM sequence, CGG. And if that CGG wasn't there, well, then this gene wouldn't be cleaved by Cas9 if this was used as the guide sequence. So very easy, right, to find guide sequences. Just look for the NGGs and pick the first 20 nucleotides, and oh, okay, you have a guide RNA that could be used by Cas9 to mutate the gene. And we could also use this sequence down here, but just keep that 5' to 3' orientation in mind, and the protospacer adjacent motif, the PAM site, needs to go NGG with the 5' end on this side right here. But we don't need to worry about that. I won't try to flip anything on you and say, look on this opposite strand on the upcoming exam. I think that's a little too confusing right now. So, okay, what next? Okay, so let's take a look at just briefly. So let's say this is our target gene here, and we want to mutate this gene. We want to mutate it because we want to see what happens. Say we're studying mice, and we want to mutate this gene and see, what happens to the mouse. What phenotype does the mouse have? Or, any organism. Well, what we would do is we would inject Cas9 plus the guide RNAs into the cells we want to mutate. Let's say we wanted to mutate the, say, a mouse embryo, or a mouse zygote. What we would do is inject Cas9 with the guide RNAs for this gene into that zygote. And what Cas9 would do, it would use the guide portion of the guide RNA, find the complementary site, and it would cleave that. So let's say we cleaved right in the middle of that gene. So we have gene 5' end of the gene and 3' end of the gene over here. Okay, so what's going to happen next in that zygote? Now maybe you can think about our section of the course on DNA mutagenesis and DNA repair. We talked about 2 DNA repair processes that would repair double strand breaks, because that's what Cas9 does. It puts a double strand break into the DNA. So it totally cleaves both DNA molecule, cuts both strands. So the cell's repair processes are going to try and repair that. And so it could be repaired by at least 2 mechanisms: non-homologous end joining or homologous recombination repair. Now, if you remember non-homologous end joining, what does that do? Essentially, it just makes the ends blunt and sticks them back together. And when it does that, it often introduces mutations. It can remove a few bases, it can even add a few bases, but it usually causes mutations in the gene. And if by some chance they should get lucky and stick it back together without mutations, what would happen? Well, probably Cas9 and the guide RNA would cut it again. And that would keep working until either there's no Cas9 or guide RNAs left in the cell, or there's a mutation here, and say the PAM is not there anymore, or the guide no longer recognizes the sequence because it's been mutated. So repair by non-homologous end joining would result in a mutation there. And okay, we've succeeded. We've mutated that gene. And perhaps, if the mouse zygote is diploid, both copies of that gene would be mutated. Now, another possibility is the gene could be repaired by homologous recombination mediated repair. And if you remember with HRR, this type of repair, what happens is there are proteins in the cell that use these broken ends to find a homologous template. So they use the broken ends and they'll find a homologous template, possibly on a sister chromatid, possibly on a homologous chromosome, and it could use that to fix this, and that's a high fidelity type of repair. So it would be fixed without mutation. And again, well, if it's fixed and there are no mutations, well, then the Cas9 and guide RNA can mutate again. So this could be repaired. And even if it is repaired, well, it'll keep getting mutated until Cas9 and the guide RNAs are gone, or until the mutation is present and Cas9 and the guide RNA no longer recognizes it. So then we have a mutated gene there. So the reason why I wanted to bring up this right here is scientists have figured out something else. So instead of just mutating a gene, well, you can actually use Cas9 and the guide RNA to make specific changes to the sequence of a gene. And how would you do that? Okay, so similar process. So here's that gene, and let's say we want to change the sequence somewhere in the middle here. Okay, what do? Well, we have to target Cas9 to the middle of that gene. So here's the Cas9 and the guide RNA. And okay, what's going to happen? Well, we already went over that, right? So it's going to cut the gene, put a double strand break into that DNA sequence of that gene. So then we have the 5' gene sequence over here, the 3' gene sequence over here. And then this is going to get repaired by non-homologous end joining or homologous recombination mediated repair. Now, non-homologous end joining, if that's used, it's probably going to lead to a mutation. If HRR is used, well, it could possibly, fix it, but then Cas9 can just chop it up again. But what if the cell had another DNA template? That wasn't exactly like, let's say, the homologous chromosome or the sister chromatid. It was a little different. It was a little different than the gene sequence, exactly at or very close to the region that was cleaved by Cas9. And so scientists have figured out that they can give, let's say, here's the 5' gene sequence that matches a little bit over here. And then here in the middle is going to be a new sequence. And then here on the other side is going to be a sequence that matches the 3' end of the gene. So here's the homologous template. So scientists have figured out they put Cas9, they put a guide RNA in there. Cas9 can cleave the gene, and if they give a homologous template, a DNA molecule that matches on either side of the break, so this 5' end matches this side, this 3' sequence over here matches this side. I should say this is emphasized. This is a DNA molecule, double-stranded DNA molecule. Then if this is used, and there's a whole lot of this template that was injected into the cell, well then homologous recombination repair will use this as a template to repair, and then this new sequence will get copied into the gene. So then we end up with a gene that looks like this, with the desired sequences right in the middle there. I'm going to call this the modified. And maybe we wanted to change a part of the gene to have certain codons, to change certain amino acids. Maybe there's a hypothesis that certain amino acids on this part of the protein, which is encoded by the gene, will make for a more functional protein. Who knows? Who knows exactly? Anything is possible here when you can make specific changes to the sequence of a gene. And so how could this happen? So let's say cells prefer non-homologous end joining over homologous recombination repair. Well, researchers are finding that it's possible to, or I should say they're looking for ways. Researchers are currently looking for ways to shut off this process, non-homologous end joining. That way the cell can only use this process to repair the gene. And if you give it the template, a lot of the template, it's more likely to use this homologous template to fix the gene than it is to use a sister chromatid or homologous chromosome. And another key thing right here, the new sequence is not targeted by Cas9 in the guide RNA. So it's slightly different and no longer recognized here. So once it's repaired, it's no longer targeted by Cas9 in the guide RNA, so it won't be mutated. So how could we turn off non-homologous end joining? Well, there are key proteins that are required for non-homologous end joining. So if you could sort of inhibit those proteins, well then this wouldn't work, and the cell would have no choice but to repair this by homologous recombination repair. Okay, so we're going to see some specific examples of how to use Cas9 and the guide RNAs to make specific genetic changes in, I think, plants and mammals in lecture 24. So I'll see you in I'll talk to you in lecture 24.